Okay, so we will move on to the next talk by Dr. Mukund Thattai from NCBS. So he will be speaking about statistical cell biology, searching through billions of possible cells to understand the evolution of complexity in real cells. Over to Dr. Thattai.
So thank you for having me at this meeting. I've really been enjoying myself. There's a lot of stuff I've learned yesterday and today, and it's also great to see a diversity of students over here. And I hope you're realizing how exciting biology actually is these days. My background is actually in physics, and I was bitten by the bio bug during my PhD, and then I totally switched over. So I'm going to tell you about a sort of approach we use to understand the evolution of complexity in cells. I'm calling it statistical cell biology. It's not really a discipline, but I hope this talk gives a flavor of that. So the work I've been doing is to understand the emergence of so-called eukaryotes. Eukaryotic cells are the cells that make up humans, plants, unicellular eukaryotes like amoeba and paramecium and so on. Eukaryotes are one of the three types of cells that exist on Earth today. The other two types are called prokaryotes. They're the bacteria and the archaea, which are actually very, very different. And then there's us at the tip of this. So a lot of evidence now tells us that 3.5 billion years ago, prokaryotes already existed on Earth. There's actually even fossil and chemical evidence for this, both bacterial and archaeal types. But somewhere between 2 and 1.5 billion years ago, a new type of cell emerges, which are the eukaryotes. So the question is, how did they come about? How are they related to the cells that existed already? And people were speculating about this for the long time, ever since the idea that several types of cells existed from the 1970s became very clear. But the answer is quite astonishing. The answer is that actually eukaryotes arose as a merger of the two prokaryotic groups. And this is actually the biggest hole in any phylogenetic tree that you'll ever see. It's two branches separated by 1.5 billion years that then refuse to make the eukaryotic group. And the way this works is actually very, very unusual. It's not a breeding or a hybridization. What happened was that bacteria, which used to live free, entered into a symbiotic chemical exchange relationship with archaea. And so this population of bacteria and archaea ended up making closer and closer physical contact over potentially millions of years. And eventually the bacteria got engulfed, not by one cell, but this is population, leading to the first eukaryote, which would be a cell that has, let's say, a nucleus and these entrapped bacteria, which are now called mitochondria. So if you look at eukaryotes today, they have genes that come from both the archaeal parent and the bacterial parent. But what's interesting is that both parents are very, very simple cells architecturally, whereas eukaryotes are obviously incredibly complicated. They're much bigger, they have much bigger genomes, they have things like sexual reproduction, they have a nucleus, Golgi apparatus, and this internal architecture. So I'm interested in how that architecture came about over this billions of years of evolutionary time. So we've done a lot of work on this over the years. So if you look at this timeline, which is, let's say, from this 1.5 billion year old first eukaryote to what's called the last eukaryotic common ancestor. Yesterday we heard about how to reconstruct the most recent common ancestor. The last eukaryotic common ancestor is the common ancestor that links you and trees and whales and amoeba. And it turns out that that cell is actually just as complicated as any eukaryote today. So the internal architecture of eukaryotes hasn't really changed much since this time point of about 1.5 billion years ago. And so that increase in complexity somehow happened on this long branch. And the problem with evolutionary biology is that if you have no intermediate states, human evolution you can reconstruct because you have human fossils. So you can actually find these intermediate forms and make hypotheses about how they connect up together. But for cells, they very rarely leave fossils. And whatever intermediate forms existed, for example, you can ask, was there ever a cell with a nucleus but no mitochondria? Or a cell with mitochondria but no sexual reproduction? You don't have any of those intermediates. You just get the whole thing all at once because all those forms died out. So somehow you have to use some other trick to reconstruct what happened on that long branch. So we've done work on the archaeal origins of eukaryotes. We've done work with deep time phylogenetics on reconstructing the earliest mitochondrial engagement with the host cell, and then more recently looking at this membrane trafficking system, which is what I'm going to talk to you about today. So this is the system I work on. Membrane traffic is a catch-all for all the things that move around inside a eukaryotic cell on these little carriers called vesicles. So vesicles aren't drawn in this picture, you'll see it in the next slide, but they move cargo between these different places inside a eukaryotic cell. A eukaryote will have a nucleus and an ER and it has a plasma membrane. This is just a cross section of a much bigger cell, and the Golgi apparatus and endosomes and lysosomes, and so these are all places, right? You saw a lot of graphs already in earlier talks. You can think of this as a graph with nodes and edges. Each node is a place, each edge is a flux of material. That material is actually carried by these small carriers called vesicles, which I'll explain in a second. What's interesting to me about this is that there's two types of emergence in this abstract problem. So one is how did this emerge over evolutionary time? It's sort of natural history or retrodiction, right? But the second question is, how does it emerge from molecular interactions? What are the molecular rules that create this architecture just from much more small scale, nanoscale protein and protein chemical interactions? So for that we have to zoom in a little more. So I told you about these vesicles, which are these carriers. You can think of a truck that moves cargo between different warehouses. So in the same way, between a source compartment, let's say the ER, and a target compartment, let's say the Golgi apparatus, you have to move proteins and lipids from one place to another. It doesn't just happen through the cytoplasm. The material gets packed into these spherical carriers, which are 40, 50 nanometers in size, called vesicles. They actually pick up very specific cargo from the source warehouse, and then some of that cargo actually tells them where to go. So it has an addressing system, right? So this little protein, that little red protein that sticks out over there is called a snare protein, and the snare protein is the addressing system. And that addressing system will then go to the target and say, do I match a complementary protein on the target? This is called a T snare for target snare and a V snare for vesicle snare. If the V and the T actually like each other in terms of some sort of physical interaction, it will bind and deliver the cargo. So what I'm talking about, the addressing system, means that there are actually dozens of kinds of V snares and dozens of kinds of T snares, and they actually decorate different surfaces in the bacterial cell, in the eukaryotic cell. And so every vesicle will know where to go by testing the sort of pairing between its V snare and a potential target T snare, and it delivers the cargo. But the important thing is that this process also requires a certain amount of recycling through the whole system, right? Once you've delivered the cargo, that snare itself has to go back. And so if you look at this system, that gives you a much better idea. Every compartment in the cell is constantly exchanging cargo, okay? It's gaining and losing, but it doesn't change its own composition, right? So somehow, despite all this flux, there's a steady state that emerges. It's sort of, again, this is a type of emergence. So we've worked a lot on many different models of vesicle traffic over time, and I'm not going to spend too much time on these frameworks, but for example, you can make a standard dynamical systems framework where you say that the chemical composition of each compartment is represented as a vector which changes over time, and these exchange fluxes change where the vectors are pointing, and you can start with random initial conditions, and then you can ask how to actually load in the right matrices of interactions between proteins so that there is a steady state that emerges which has a certain number of compartments and a certain sort of flux between them. I'm phrasing this as an inverse problem. Actually, what we do is a forward problem where we say, let's take a matrix and see what topology of the trafficking network emerges. I'm going to spend a little bit of time today talking about a slightly more discretized version of the same problem, where we think of the system as, again, a graph with nodes and edges rather than a big continuous chemical space. And we also think of the composition as discretized, that is, it either has some chemical or doesn't have some chemical. And so each compartment is then represented by some sort of Boolean string, and each vesicle is represented by a Boolean string, which is a composition string. And there's some sort of, again, molecular interactions that determine which compartments give which vesicles can fuse to which compartments. And this then results in a topology of a vesicle trafficking network. And again, and this I'll spend a little bit of time on. What I'm not going to talk about is also very recent work where we realized that after doing this, the theme of today's talk, which is screening billions of cells and seeing the topologies that arose, we realized that there were very consistent topologies that kept on coming up through these random sampling methods. And it turns out that these topologies are not accidental. If you want a vesicle trafficking system, which is Golgi, ER, plasma membrane, lysosome, whatever it is, with vesicles that couple them, and if you want to ensure that every vesicle can correctly fuse to every target, the compositions of these vesicles are very important. They have to be sufficiently different that they don't make mistakes. And it turns out that the answer to this question can be framed purely in terms of the topologies of these graphs. So for example, there's something called Menger's theorem, which we used, is from the 1970s, which says that if the graph is 3 strongly connected, it's a strongly connected graph, and it has at least 3 edges across every cut, that is sufficiently complex to admit a vesicle trafficking solution. Okay, so it's a very nice topological description of a very complicated chemical requirement. So I'm not going to talk about that today, but you can read about that here. I am going to talk about a little bit about these Boolean networks before going on to the main topic of today's talk, which is the evolution. And this is just to give you a flavor of the kind of work we do. So the Golgi apparatus, you've seen it in textbooks. It's a remarkable organelle, and I'm not going to tell you much about what it does, but it's a factory that creates complex molecules. And these molecules move from one workstation in the factory to the next, like a production line where you make a car in a factory, and then exits to the surface of the cell where this complex molecule is used for cell-cell interactions. So the Golgi apparatus is actually a very dynamic organelle, and it turns out that this picture of the Golgi is wrong in textbooks where you think that the cargo is moving from one place to the other. In fact, what happens is that each one of these little plates or cisternae stays fixed and matures over time. Its composition actually changes from this light blue state to this dark blue state. So it's not that the car moves to the factory, but the car stays in one place and the workers keep moving backwards through the production line and they finish making the equipment, which is also a nice way to do it. And so you can make these dynamical systems models and you see the correct oscillation patterns which represent the movement of the composition from this chemical type to that chemical type over time. There's a sort of oscillation in this dynamical space. But what we realized was you didn't have to have such a very complicated model to describe the Golgi apparatus, but a simple topological model was enough where you create the first compartment, the compartment matures over time from one chemical state to the other because the chemicals from here are being sent back to here and back to here. And so this little description captures in a nutshell what's happening in a much more complicated stochastic sort of Markov space. So we said, why not just use this description for the whole thing? So we made a little model where we said that you have vesicles and compartments described by Boolean strings, and you say how many descriptors there are. So if there are n descriptors, there are 2 to the n possible compositions. And for each of these compositions, you have to say, can that vesicle give that, can that compartment give that vesicle? Can that vesicle fuse to that compartment? So we call these the budding and the fusion matrices. And you have to fill these based on some knowledge of how the molecules work. And since we don't know how the molecules work, we fill them at random. And we hope for the best, and we see what happens when you sample many, many random kind of matrices over here. I mean, they're not really matrices, they're graphs, okay? And so you can take these molecular rules, you can initialize the system with some number of compartments, which make vesicles, which fuse to the compartments, which change their composition, run over time, reach a steady state, and then see what the steady state topology is. And the upshot of all this is that if you take random budding and fusion rules, where I pick these ones and zeros based on a statistical density, let's say, and you can sample thousands, hundreds of thousands of these, then there's certain types of compartments that arise and certain types of vesicles and certain types of topologies. And it turns out it's not so hard to find a topology in this space. So you take one of the dots here, every dot is a sort of potential synthetic cell. And it's easy to find synthetic cells that look very much like real cells. This real cell has the ER, the cis medial trans Golgi, a plasma membrane, early endosome, and so on. When I say they look like the real cell, I mean statistically. They have the same number of maturation chains where one compartment becomes another, roughly the same number of vesicles per compartment, this kind of thing. And we actually can say more. We can actually say that certain motifs arise disproportionately, and we sort of understand why that happens, though I'm not going to tell you about that today. Okay? So that's one aspect of statistical cell biology. You make random molecular rules, and you see that of this selection of random molecular rules, what outcome do you get? The outcomes turn out, because of the constraints of feedback and recycling and so on, the outcomes turn out to be very non-random subsets of the network topology space, even though the molecular rules are random. I mean, it's not surprising, but it's nice that you can actually make such a connection. So then we went back to our primary interest and said, well, how do we bring evolution back into this? So that model had no evolution in it, because honestly it had no molecules in it and no genes and no mutations. So we needed a mathematical model that had all these features, genes, which can mutate or duplicate or get deleted, and modules of proteins that interact and can be regulated, which are encoded by these genes obviously, which then result in trafficking networks that have compartments and flows between them. This determines that. And then some notion of function that you can select on to really close the loop here. Because when you're typically doing abstract evolutionary biology, you just assume that there's some fitness function. But here we wanted to have an explicit fitness function for a vesicle trafficking network. So it took us a long time to do this. And the trick turned out to be, well, maybe I'll tell you the trick in the next slide, but let me just tell you what the framework is, okay? And you can actually do this on the back of a napkin, it's not very complicated. So in this model, there are compartments which are these big nodes, and there are these vesicles which are these small nodes. And then there are coats, okay? So a coat is the thing that makes a vesicle and chooses the right kind of cargo to load on this vesicle. It's a sort of manifest for the FedEx guy to come and pick up the right cargo. And there are some number of coats. So in our cells, there are some 7 or 8 kinds of different coats. But let's assume a primordial cell that has only 3 kinds of coats for the sake of simplicity. These 3 coats are red, green, and blue. any cargo in the cell will either like or not like each one of these coats. So it will either get on the red truck or the blue truck or the green truck or some combination. So therefore, abstractly, there can only be 2 to the 3 is 8 kinds of cargo. This one doesn't get on any truck at all. This one gets on every truck indiscriminately. This kind of cargo only gets on to the green and the blue trucks and so on. They're colored in the obvious coloring convention. And so every vesicle can only contain a certain subset of cargo. Of course, it also has to have it on the source compartment. Every compartment can also be labeled by the kind of cargo it contains. Now I go to my cell biologist friend down the hall and I ask her, tell me what your cell looks like as far as you know. So she'll draw some sort of diagram saying, this is the ER and here's the vesicles that think move between these compartments. And then I'll say, well, can you at least commit to what coats are causing these fluxes to happen? And they'll say, yeah, some genetic evidence and knockout and CRISPR and so on tells us that it's the red coat that does this and the blue coat that does this. Okay. Once I have this, it's an immediate single back of the envelope calculation, or just it's very, very simple to say that this must be the composition of such a cell. And the trick over there is to realize that the cell actually makes every protein and lipid in the ER. So all you have to do is put every type of cargo in here, let it flow and see where it ends up. Okay. So remember this, there's no molecules here at all. Here somebody's told me the solution and I say this must be the composition. But now that composition must have a molecular explanation. Why is it that that vesicle fuses here? Why is it that that vesicle buds out from here? And so here's where the molecular rules come in. I have to assume that molecules have some sort of function. And so here it becomes very, very specific from the cell biology, right? So I then put in very explicitly molecules that are known to biologists and biochemists. There are recruiters that recruit coats to the compartment. There are V snares and T snares that are these pairwise matching addressing system. And in this particular type of model, there is an activator of a V snare. Turns out V snares are regulated. So each of these is a kind of very specific molecule. And every one of those molecules can be one of 8 cargo types. Okay. Now just take a look at this question. Why is it that the red coat buds out from this compartment? I can hypothesize that it likes the cyan recruiter, and that's why it buds out from there. And then somebody will say, well, does it bud out from every place where there's cyan? And it's true, it does. So this is a sufficient explanation for those 2 vesicles. Why does this vesicle fuse here? I can say that, well, a magenta V snare activated by a yellow will fuse to a red, and you check and that does happen. But I have to play this game for every one of the things that happens here. Turns out I can, and here's, for example, a full molecular solution to make this work. This was the trick which I mentioned earlier. In my previous work, I said, here's a molecular rule, what network arises? We flip the problem around. Here's a network, can you find a molecular solution? Right? And this turns out to be the thing that really makes us go forward in this. I also, and I'm not going to talk about this very much today, introduced the idea of mutation, that a cargo can change its type or change its color over time due to mutations in the tail region that is cytoplasmically exposed. Putting all this together, so now I have a network which is a hypothetical network. Somebody said, here's a network. Here's a potential molecular solution. But that's just one molecular solution. Any one of these boxes, could be occupied or not occupied. In the genome, such a protein could exist or not exist. So it turns out that in this very simple model, there's like 536 possible kinds of molecules the genome could encode. Either it has it or doesn't have it. So you fill these with ones and zeros. And for any given network, it turns out that it's like a Boolean satisfiability problem. There's a certain set of ones that can never occur, forbidden places, and then there's a certain set of places where you have to have at least one in those bubbles. And if you have that, it's a molecular solution for this molecular problem. Okay? And if you put all this together now, it turns out that, and here the red, green, and blue doesn't have anything to do with the coat colors, I'm sorry. Here, red, green, and blue are different types of vesicle trafficking network topologies. And I can say what in the space of all possible molecular repertoires, which is huge, it's 2 to the 536, what are all the ones that give the blue type network? And it turns out that there are many. The same network can have different satisfiability, Boolean expressions, which we call covers. But anyway, there's one network with many possible solutions. Here's an interesting place where you can make either a red network or a blue network with the same set of molecules. It's a sort of type of bistability. So you can actually evolve from one to the other if you want to. Here's a more interesting place where one type of network region kisses another type of network region. All you need is one mutation to kick you over. And the most interesting is that there are some networks that are just not possible. There's a requirement, something has to happen in a zone where something cannot happen. It's a contradictory thing that has no solution at all. So these are the impossible ones. So if you actually test all this, so then what we do is just very, very simple. We just comprehensively check every possible network topology up to a certain size. Certain number of nodes, certain number of edges. So here's the number of nodes and edges. It explodes obviously. But it turns out that very few of these have solutions, especially when you get to big networks. You can test billions of networks and for a certain type of molecular activation rule, you get very, very few solutions. And it's not surprising because the bigger the network is, the more things can sort of conflict with each other. You just don't find a solution, but you do find. Okay? So now we take the networks that have solutions, and then we ask, can you change one network, which I know has a molecular solution, to another network, which I know has a molecular solution? In this case, for example, both networks have the same molecular rule, but that's not the important thing. But evolutionarily, can you talk about function? Okay? So here, this is a place where, you know, all so somehow evolutionary biology has been removed from biology because nobody can decide what a cell actually wants to do in the environment. It's a very difficult empirical question. So this is why, you know, there's a lot of very abstract evolutionary biology principles about, you know, fitness landscapes and things like that. But there are very few cases when you can bring a very detailed model like this and say there's some notion of selection. What did the proto-eukaryote want to do? One of the things that it wanted to do, it turns out, is it wants to do phagocytosis. It wants to bring in food using an endocytic pit and digest the food internally. The other thing it wants to do, like the Golgi, is make complex molecules, secrete it and expose it to the cell surface. But these are all sort of units of function which are too detailed for me to incorporate in an abstract model like this. So instead we said, well, can we select on something called an affordance? So affordance is something that comes out of, you know, for example, user interface design. And affordance is something on an object that tells you how to use that object. So for example, on this bottle, the cap is a different color, which sort of signals to me that I need to do something with the cap to open it. Or on a door, if you have a door handle, it means pull. If you have a plate, it means push, right? So these are things that make it obvious how to use an object, even if you don't know any underlying theory. So what does that mean in this case? Well, so imagine you have a vesicle trafficking network, you have compartments, you have edges, and now I have some complex function where I want to deploy proteins across different compartments in the cell and have them move around. What would make it easy for the cell to do that? So one thing that will make it easy, so again, think of warehouses and trucks, so in this case, like bus stops and buses. It's nice if a bus stop has an easily recognizable color, right? So that I can always just show up there and just, without doing too much computation, know that I'm over there. So in this case, for example, the red label is only on this compartment and not on that one. So it's very easy to distinguish, and the black one is only here and not there. There's many others, but, you know, for example, these two labels. And for example, it'll be useful to know that if I get onto the pink bus from the red bus stop, it'll only go to the black bus stop and not go in, you know, unlike in Bangalore, let's say, where you don't actually know where the bus is going, right? So it's nice to have labels on the edges. And then I just require a very simple thing. If the network changes, I just require that the new network has at least the same set of functions as the old network. Because by this time, the cell has gotten used to a certain lifestyle, and certain essential functions depend on the idea of a red bus stop and a pink bus and a yellow bus and so on. So in this case, this is actually true. Both these networks, if you check very carefully, have exactly the same functional motif, right? There's only red and black and this edge, because cyan is ambiguous. It's on 3 different places, and in this case, yellow, you know, between these two is ambiguous because it's going both ways. So this is the notion of function. Here's an example where it doesn't work. Here I have function. Here I have more function. Actually, this network has the old function and much more, which means you can go forwards, but you can't go backwards. It introduces the notion of a ratchet. Okay? So I don't want to spend too much time on this, but what I've brought now is the idea that I can have selection, which is a sort of unidirectional thing. It's not always that selection selects for complexity. Okay, that's not what I'm saying. All I'm saying is selection ensures that if there's a set of functions, that functions are at least maintained over time. There is a ratchet, so but it's a subtle difference between having more complex structures and it's having more functional structures. Okay, so now that I've done this, and I remember I already have my hundreds or tens of thousands of networks for which I know the molecular solutions. So now I can ask, how are they connected by mutations and how are they connected by selection? So every little dot here is actually a network with a molecular solution or a family of molecular solutions. Every edge between them is either a mutation or a non-mutation, that is a bistable system where the same molecular solution works for both, which allows the cell to move between both these states with very little genomic change. But I have tens of thousands, so I have to build an entire graph. All the graphs in this little bubble, it turns out, have the same functional motif. But I can go from graphs that have one type of functional motif to a more functionally capable motif. So for example, 2 compartments and 1 edge to 3 compartments and 1 edge. In that case, evolution will tend to move you in this direction and less likely to move you backwards. So I take this graph, zoom it out to this level, and now here what I've shown is a very coarse-grained picture of the same thing, where every one of these little, so this here is, each one is like an island which contains thousands of little networks, all of which have their own molecular rules. Inside, of course, you can keep moving around, but you don't increase in functional complexity. But sometimes you have a mutation that takes you to a more complex island, that is, it has more edges and more compartments, one or the other or both. And then you hang around there for a while because you're just moving around this neutral space, and you keep going. Right? And then now you can actually simulate evolution. This is meant to be some sort of proto-eukaryote. This is meant to be a complex cell like present-day cell. Not really, but let's say at least more complex than the ancestor that you started off with. And you can say what flows will happen in this kind of topology. So I'm not going to tell you everything we know, but I'm just going to highlight a few things. One is that this is very interesting. Remember, I had this huge molecular space where almost nothing actually made a functioning vesicle trafficking network, right? Trafficking networks are very rare in molecular space. So you might imagine that it breaks up into a bunch of disconnected pieces evolutionarily. But it turns out that although solutions are rare, around a solution, there's a bunch of neutral changes you can do. So there's a very large island in which the same solution works. And then it turns out that these islands are actually connected. So although solutions are very rare, the entire system is connected mutationally, which was surprising to me, but it turns out to be the case. That's the first thing. Second thing is that there are actually dead ends in this evolutionary pathway. It turns out if you end up in this system that has two compartments at two edges, you can never get out of there for some reason, right? There's no mutation that will get you out without going back down and walking up again, which of course can happen with less probability. It also turns out that there's a funneling effect where one type of topology is selected disproportionately over all the others. And remember, there's so much complexity going on underneath. So this is also very surprising that it selects for this sort of very, very few kinds of ultimate topologies. Again, it's a reflection that certain motifs are much more likely to happen under these constraints of recycling and so on. So to really wrap up over here, what we have is a fake.
So you have 2 minutes more.
Yeah, this is the last slide. What we have is sort of a fake picture of what happened on that 1.5 billion year old branch from the first eukaryote that just had a plasma membrane, sort of one compartment, to a system where the archaea and the mitochondria got together and had some primordial internal membrane. We don't know what it did. We don't know if it was derived from the PM or derived from the nucleus, but it is the first time there's an intracellular compartment that does something interesting like phagocytosis or secretion. And over time, the system gets more and more complicated through this sort of ratcheting effect. So the questions you can ask are manifold. You can ask what kind of molecular changes tend to happen on the most common branches over here, and we do see that. You can ask a very interesting question. You can ask, did the first intracellular compartment derive from the cell surface or derive from the nucleus? And it turns out there are many more solutions where it derives from the cell surface than from the nucleus, which is again unexpected, and now we sort of understand why that is the case. And we can ask, at what point in time does the system actually reach a point where it is complex enough to represent a real cell? This is interesting because it turns out that if you have a 2-coat system and not a 3-coat system, remember I had red, green and blue, if you just had red and green, you can't make anything as complex as a real cell. So there's a threshold of, in this simple model, 3 coats that needs to be there to have a certain level of complexity. So the emergence of the molecular repertoire precedes the emergence of cellular complexity in this case. Of course, I can add more coats later and different kinds of activation, inhibition and so on. This particular motif is very interesting because it contains every kind of thing that people have been saying proto-eukaryotes need. Phagocytosis to eat, endocytosis recycling to signal and sense the environment, and modification and secretion to decorate your cell surface for sexual reproduction and cell-cell contact. Okay, so here's the lessons we have from all this. So what do we know so far? Right? None of this is really testable in the lab. Okay, so we'll say that right now. But it does resolve some puzzles. One puzzle is that all eukaryotes today have the exact same vesicle trafficking system, the topology at a coarse level. There's been no change in that system for 1.5 billion years, although there was a huge amount of complexity increase from the proto-eukaryote to that. So why was there such a huge phase of increase and then a static description after that point? And the idea is that once you reach a certain level of complexity, it's hard to go forward. So these molecular constraints lead to these constraints on vesicle traffic topologies. There's also high redundancy. I can't use this system to actually tell you which molecular rule is happening for which topology, which any cell biologist will say is realistic. How much time does it take? Well, it turns out these trajectories, I didn't tell you, but they're dominated by exponentially long neutral walks. You're sitting in a little island of a Markov chain where you spend a lot of time before you exit. Okay? And there's no simple link between genes and new compartments, so I can't really do phylogenetics on the genes to guess what the compartments would be. So in some sense, it's telling us the limitation of a protein or a phylogenetics-based approach for eukaryotic reconstruction. So I'll finish with this. So the idea, actually this is borrowed from this thing called assembly theory, which I'm actually not using, but this picture is very nice for me. Assembly theory is a way to think about the increase in complexity in molecules. I'm talking about the increase in complexity in trafficking networks. I can write down topologically, all topological trafficking networks. This is the universe of all mathematical possibilities. I can talk about those that are chemically possible. Of those, I can talk about those that are contingent. Contingent means reachable by evolutionary steps. And finally, those that are observed. And I think we really can make a claim that there's a lot of filtering going on here that explains some of the reproducible features of today's vesicle trafficking systems. So I'll stop over there. You can read more about it over here. So this is work by many students over many years in the group, but most recently the work I've shown is the work of Sahana, who's a graduate student in the lab. So I've been working on this, we framed this problem in 2010, very long time ago, and it's taken us this long to come up with a nice framework to actually study this. So thank you for your attention, and I'll take questions.
Thank you. We will take a couple of questions.
Very insightful talk. Just curious that I know you said that you cannot just correlate the genes to the compartments, but this model at any level, does it conform to the nearly neutral theory of molecular evolution?
No, no. So this is molecular neutrality is totally different. Here we have. So when I say neutral over here, it's functionally neutral, in the sense that the network carries out the same function, but in a detailed way, it's totally non-neutral. You get new arrows, new edges, and so on. That's one point about how I use the word neutrality. But secondly, the neutral theory of evolution has to do with the statistics of observed mutations at certain population sizes and so on. This is not population genetics. Okay, I'm only giving you the evolutionary landscape. On top of this, now you have to do population genetics. Okay, so the question doesn't even arise in this context. Does that? Yeah.
Hello, sir. Here, sir.
Yeah.
So, interesting talk, sir. So, I have one question. So, I model prokaryotic organisms where, I have just a single compartment and a model. One major challenge that we face is that when you make a kinetic model involving ODEs, you have to estimate the kinetic parameters of the numerous reactions. So, when I see you have also made it for small compartments, how do you overcome or handle this challenge?
Thank you. So I flashed earlier different ways that we model these systems, right? So there are some models of ours that have lots of parameters, okay? And those models are big ODE models or stochastic ODE models, which will then result in some sort of steady state. Now I step back and I coarse grain the steady state, because just to say there are nodes and edges. And this model works at that level of coarse graining. The way to connect those two models is the claim that these topologies I'm showing, I claim should be the steady states of some bigger ODE model with some parameters. But I don't know what the basin size of it is in terms of what parameter regions and so on. So all that is left out of here. But at least the constraints we see here should apply to that also. So I can rule out an entire family of models by saying the steady states don't even apply. But the reason we started along this is because of this explosion of parameters. Yeah.
Okay. Thanks.
Yeah. I don't know who's got the mic. Oh, yeah.
Okay. So, very nice to see how it works. You mentioned that I understand your interest is in the mutations in evolutionary perspective. But how does the model work with respect to, say, mutations that are disease associated? What kind of mechanistic insights does it give?
Yeah. So it's a good question. So over there actually it's much easier. So it turns out that then I have a nice constraint space where I can say that if you have this vesicle trafficking system that you claim is doing whatever you think it's doing, I can say that there are missing edges. This doesn't actually work with recycling. This doesn't work with fusion. So it works very nicely. The problem is that nobody ever has the complete information network, I mean, not even close. So this idea that I'm going to tell you there are missing edges, of course there are missing edges, and there are too many potential ways to solve those missing edges for me to make a prediction. Okay, so the kinds of models we use in collaboration with experimentalists are not these. We make a different set of very detailed models that can then be fitted statistically and so on. That's another half of what the group does. It's not this.
Thanks.
Very interesting talk. So, couple of questions. You talked about evolutionary trajectories coming to dead ends, right? In the previous slide. This one, yeah. So now, in your whole landscape of all the solutions that you had, did you had any organisms that were missing any one of these and were still viable in one sense, some sense?
Yeah. So I don't say anything about viability. Everything that
I mean to say, like, was still whatever you're calling as like
Yeah, so all of them are functional and possible. When I say something is not possible, I mean it's not even possible to make, not that it's alive or dead. Every one of these things is alive. What is true is that many motifs you could imagine are totally missing. It's the other way around. So there's a whole set of excluded motifs that you don't see over here. On top of this, of course, issues of viability arise. Finally, these are not dead ends. If I have more molecules in the genome, I can go away from this also. And there's a whole range of more complex things that happen above. So there is much more to this world.
Order of complexity.
Right. So what I assume will happen is that your molecular repertoire then determines where you end up for a long time. Then you have a duplication, and then you escape from there and go up further.
I was going to ask, have you done duplication in C?
Oh, everything is duplication. So I haven't mentioned it, but all my mutations allow duplication and deletion and mutation of genes. That's all happening underneath.
But what about, like, let's say there are cases of whole genome duplication?
Yeah, so we've actually studied that separately, and there are cases of eukaryotic whole genome duplication. It's easily put into this model because all it does is it adds a lot of ones and zeros at once, and then you can watch them move. It doesn't, so we thought you'd need, okay, just very specifically, we thought because of the sparsity of solutions, you would need some clever trick like whole genome duplication to jump in complexity. It turns out that single mutations is enough to connect the whole space. So whole genome duplications merely increase the rate of that, which is interesting, right? But unexpected.
Okay.
Yeah.
I have one question. So at the end you mentioned that it's very hard to test this theory with experiments, right? So, but you mentioned about some criteria which can be observed through experiments, like the bottlenecks that you explained, or the transitions between the islands. So whether these transitions can be, the probability of the transition from one island to another can be correlated with time scales, and then you can experimentally test it. Like
so not time scales specifically, okay. And look, the chance of actually doing any of these steps in the lab is 0. Because the space of possible mutations you have to keep on searching over to actually leap from one state to another is immense. This is what I mentioned about these long neutral walk. What is testable though? It turns out that if you have a different way in which snares are regulated, either they're activated or they're inhibited or they're unregulated or whatever it is, these are assumptions about how molecules work, which the biochemists still haven't worked out. It turns out that each one of those models gives you a different set of allowed topologies. In principle, if I had full information, I could use the topology to tell you the regulatory model. If I had full information. I don't have full information. That's the problem.
So I was thinking about an alternative approach, like whether you introduce an selection pressure in the eukaryotic cells. The eukaryotic cells having very lower replication time, and then you can propel it to move to a certain direction.
So this is, this is the experiment to do. So people have been doing a lot of benchtop evolution experiments. So one has the trick with this experiment is exactly what you said. You have to set up a selection pressure where it is useful to have more compartments. Okay, that's very subtle, and where it's not harmful for anything else. So the trick is to arrange that. If anybody is in the mood to talk to me about how to set up such an experiment, we should do it, perhaps in yeast or something where the genetics is easier.
Okay,
thank you for that.
I would like to ask one naive question maybe. So you're talking about Golgi apparatus. I was thinking that are you looking at the exosomes also?
No, we're not looking at exosomes.
Because that's also a very interesting place to look into how the things are moving there.
Yeah, so so exosomes are sort of inside the inside. If you can make this multivesicular body and then it can secrete.
Right.
So in principle, it falls within this framework.
Right.
We just haven't looked at it in detail.
Okay. So let's thank the speaker then.
Thank you.
I think you can interact over lunch. Okay?